Hello everyone and welcome to our module on the placenta. The placenta is the structure that provides nutrient and gas exchange between the mother and the fetus. As you may know, the baby cannot eat for itself in the womb and it cannot breathe for itself, so it depends on the placenta to get oxygen and other nutrients. When an embryo implants in the wall of the uterus, the uterine lining undergoes what's known as the decidual reaction. This is the reaction or change in the endometrium at the site of implantation. Decidua is the term used to describe the altered uterine lining during pregnancy, and there are a couple of subtypes of decidua that you can find in the womb during the time of pregnancy. First of all, there is the decidua basalis. It's also sometimes called the decidua placentalis. This is the uterus at the site of implantation. It will interact with the trophoblast and lead to development of the placenta. You can also find in the uterus the decidua capsularis. This is the decidual layer that surrounds the fetus. And finally, there is the decidua parietalis. This is the decidua opposite the wall of the uterus. All three of these subtypes of decidua are shown in this drawing on the screen here. So this is the decidua basalis right here. It's at the site of implantation where the placenta will form. This is the decidua capsularis. It surrounds the fetus. And then finally, on the opposite wall, you can find the decidua parietalis. There are two membranes that surround the fetus in utero. The first one is called the amnion. The amnion is the inner membrane that covers the fetus, and it holds the amniotic fluid and it protects the embryo. And if you look at this picture at the bottom right side of the screen, this shows the embryo in the bilaminar disc stage before the germ layers have formed. This is a very early stage of development, and I talk about this stage in more detail in the video on embryogenesis, but I just want to point out to you that even in this very early stage, the amniotic cavity is already present and the amnion is beginning to form. The second membrane is called the chorion. This is the membrane that surrounds the amnion and embryo. It derives from the trophoblast to support the fetus and the amnion. And if you look once again at this picture at the bottom right, the trophoblast is this layer of cells around the outside here, and it will develop into the chorion. When we talk about the placenta, there's some terminology you need to be familiar with. When we refer to the basal plate, we are talking about the maternal side of the placenta. This is the side that is in contact with the uterine wall, and it includes the maternal decidua basalis layer that we talked about earlier. When we refer to the chorionic plate, we're talking about the fetal side of the placenta. This is in reference to the chorion membrane at the level of the placenta, and this is what gives rise to the chorionic villi, which are very important structures for nutrient and gas exchange that we'll talk about in a minute. Now let's talk about the trophoblast. In the video on embryogenesis, I talked about how early on in embryogenesis, a structure called a blastocyst formed, and that's what's shown on the screen here. This is a fluid-filled structure that contains an inner cell mass that will ultimately develop into the embryo. This structure also contains an outer layer of cells called trophoblast cells, and this outer layer of the blastocyst is called the trophoblast, and that is what will develop into the placenta. The way this happens is the trophoblast proliferates into two cell layers. The outer layer is called the syncytiotrophoblast. This layer invades the endometrium with finger-like projections called villi. It also forms little spaces called lacunae, which will fill with maternal blood that will participate in gas and nutrient exchange with the baby. The inner layer of the trophoblast is called the cytotrophoblast. This layer is constantly proliferating, and cells migrate away from the cytotrophoblast and into the syncytiotrophoblast. I'll show you a picture of this in a minute. In addition, the cytotrophoblast secretes proteolytic enzymes that aid in invasion of the endometrium. And then the two structures together work to form chorionic villi. These are finger-like projections that will contact with maternal blood and participate in nutrient and gas exchange for the baby. And here's a picture to make this more clear. So we've got the uterine structures like vessels and glands up at the top, and we've got the mesoderm of the developing fetus down at the bottom. So the trophoblast extends out in these finger-like projections into the uterine structures. The trophoblast has an inner layer, the cytotrophoblast, and the outer layer, which is blue in this picture, is the syncytiotrophoblast, and they make these finger-like projections, which are the chorionic villi. The villi also leave these spaces, which will fill with maternal blood coming from the uterine vessels, and that maternal blood will give oxygen and nutrients to the baby. And here's another picture just to make this very clear. The chorionic villi are very important structures. They are basically the digestive tract and the respiratory organ for the baby, so that the baby can get nutrients and exchange gases. The outer layer is the syncytiotrophoblast, the inner layer is the cytotrophoblast, and then the chorionic villi serve as the contact area with maternal blood. Once the villi are formed, the fetal mesoderm will invade the villi. The mesoderm is shown here in my picture. It's the structure shown in red. It will grow into the villi like this, and branches of the umbilical artery and vein will form. So what's basically happening here is the maternal blood is filling these lacunar spaces. The baby's blood is flowing into and out of the villi, and that's how gas and nutrient exchange will take place. Eventually, these arteries and veins in the villi will connect to the umbilical cord, which has 2 umbilical arteries and 1 umbilical vein and carries blood back and forth between the placenta and the baby. So let's talk about the circulation in the placenta in a little more detail. On the mother's side, the endometrial or spiral arteries deliver blood to those villus spaces, and then from there blood drains out through the endometrial vein. On the baby's side, the 2 umbilical arteries deliver deoxygenated blood. Even though it's called an artery, in the baby it carries deoxygenated blood. You can think of it kind of like how the pulmonary artery in the heart carries deoxygenated blood. That umbilical arterial blood goes to the chorionic arteries and from there into capillaries which fill the villi. The capillaries then drain to a single umbilical vein which now carries oxygenated blood back to the baby. It's important for you to understand that the placenta acts as a barrier. There is no direct mixing of maternal and fetal blood under normal circumstances. Oxygen and carbon dioxide can freely diffuse in and out of the maternal blood and through the trophoblast tissue. There's facilitated transport of glucose. That's how the baby gets glucose. There's also active transport of amino acids. IgG antibodies from the mother can cross the placenta, but not IgM. And then other nutrients, drugs, and infectious agents can potentially cross. It depends on the specific thing you're talking about. And this is why some drugs are teratogenic and some are not. It's why some infections are dangerous in pregnancy and others are not. It all depends on whether or not they can cross the placental barrier. Now let's talk about the umbilical cord. The umbilical cord is a connection between the embryo and the placenta, and it derives entirely from the fetus. There is no maternal contribution to the umbilical cord. And it contains the umbilical arteries and veins, and it's what shuttles blood between the baby and the placenta. And it's derived from 2 embryologic structures. The first one is the yolk sac, which is a cavity or sac formed in early embryogenesis, and the second one is the allantois, which is an outpouching of the hindgut. So here's a very early stage in embryogenesis. We talked about earlier how you can see the amniotic cavity already, even early in embryogenesis. You can also see the yolk sac at this point in time, and this will eventually contribute to the umbilical cord. Here's a drawing that I use in the video on GI embryology because it shows all the elements that develop into the GI tract, including the foregut, the midgut, and the hindgut. All of these elements derive from the embryonic yolk sac, but at this stage of development, we only call this little outpouching here the yolk sac. The other portions of the former yolk sac have other names. And I want to use this drawing now because it also shows all the elements that will develop into the umbilical cord. So to understand this, you've got to imagine the placenta is over here on the left side of the screen, and the embryo needs to develop a connection to the placenta. Part of that connection will come from the yolk sac here. It will grow in this direction, and it will obliterate and become part of the umbilical cord. Now, just an aside here, if the yolk sac fails to obliterate, that can form a Meckel's diverticulum, which I talk about in the video on GI embryology. The other element of the embryo that will grow into the umbilical cord is the allantois, but before we talk about that, let's talk about the cloaca. So this bend down here is the cloaca, and it will connect to the outside of the embryo. It will then divide in half, and the bottom portion will become the anus, and that should make sense to you because that inserts into the GI tract here. The upper portion after the cloaca divides is called the urogenital sinus, and I talk about this structure in the video on genital embryology. It develops into a number of internal structures, including the bladder, which will form right about here. And if you understand that, then it's very easy to understand what happens to the allantois. It grows in this direction and combines with the yolk sac to become the umbilical cord. The walls of the allantois will become the umbilical vasculature, and then the allantois will obliterate and become a fibrous connection called the urachus, which connects to the dome of the bladder, which sits right here, the point at which the allantois grows out from. And here I've summarized some of the important points. The allantois is an outpouching from the wall of the gut. The walls of the allantois form the umbilical blood vessels. The lumen occludes during development, and it becomes the urachus, which is a fibrous remnant of the allantois, and it will connect from the bladder to the umbilical outlet of the embryo. In utero, it will continue into the umbilical cord and go to the placenta. So now that we understand how the umbilical cord develops, let's talk about the structures in the umbilical cord itself. A normal umbilical cord has 2 umbilical arteries, which carry deoxygenated fetal blood to the placenta, as I talked about before. The normal umbilical cord has 1 umbilical vein, which carries oxygenated fetal blood from the placenta. And when you do your rotations in OBGYN, one of the first things you will do after you deliver the placenta is take a look at the umbilical cord to make sure it has 2 arteries and 1 vein. Also shown in this picture is an allantoic duct, which is a remnant of the allantois that can sometimes be seen. There's also all this white stuff, which is called Wharton's jelly. And the reason you count the vessels after delivering the placenta is because a single umbilical artery is an abnormal variant of the umbilical cord. It's often identified on prenatal ultrasound, but if it's not, you can identify it after delivery. And it's associated with fetal anomalies, including many types of aneuploidy and other congenital malformations. It doesn't necessarily mean there's something wrong with the baby, but there's a higher likelihood of the baby having a problem if there's only one umbilical artery. Other structures you can see in the umbilical cord I mentioned before. One of them is Wharton's jelly. It contains mucopolysaccharides. It's very similar to the vitreous humor you can find in the eye. And you may see the allantoic duct. This is the structure that connects the fetal bladder to the umbilical cord. It obliterates in development and becomes the urachus, but sometimes the duct can still be seen in the umbilical cord, as is shown in this picture up here. So now let's talk about the urachus. This is the remnant of the allantois. It's a connection between the bladder and the umbilical cord. In the adult, it is a ligament called the median umbilical ligament, and it's associated with adenocarcinoma of the bladder. Adenocarcinoma is a very rare type of bladder cancer. If it does occur, it will usually occur at the dome where the urachus inserts. And there are a number of congenital urachus anomalies, but they're all easy to understand if you appreciate that the urachus is a hollow tube in utero that should obliterate. And if it doesn't obliterate, you can get any of the anomalies shown in this slide here. So first of all, you can have a patent urachus. This is where it fails to obliterate. And in this case, there will be urine discharge from the umbilicus because you have a tube connecting the bladder to the umbilicus. Another anomaly is called a vesico-urachal diverticulum. This is a diverticulum of the bladder. Basically, it's just an outpouching of the bladder because the urachus did not completely obliterate. And then finally, you can have a urachal cyst. This is what happens when there's partial obliteration of the urachus. A fluid-filled cavity can form filled with urine. It can become infected, and that's called a urachal cyst. I'll finish with this slide on a topic that is very high yield for the USMLE Step 1 exam, and that is the immunology of pregnancy. So the fetus contains foreign antigens compared to the mother. Half of the genes in the baby come from the father, and therefore the baby can express different HLA proteins compared to the mother. This could elicit an immune response from the mother, but the baby is protected by the placenta. There are several mechanisms that have been described to explain this protection, but one of them is that trophoblast cells have been shown to not express many MHC class I antigens, and it's very high yield for you to know this for your exam. There are also studies showing that the placenta secretes substances that block the immune response, and there are probably other mechanisms as well. But it's very important that you understand that the fetus has different antigens compared to the mother, and that the placenta protects the baby from maternal immunity. And that concludes our video on the placenta.